discuss a little bit about what we do at the SIBYLS with SEC-coupled SAXS and multi-angle light scattering. We'll mention a similar way, as Greg mentioned, a few projects that we've done in the past year, and where we go with the high throughput SEC-coupled SAXS, how far we are with the users projects, and how we also delivering the data. Probably in the next cycle will be a little bit different. For those that use the SEC-SAXS at the SIBYLS, we'll need to adopt a little bit, and I think it will be a smooth transition. Generally speaking, I can move my slides. Yes. So this is our current system. We already, Greg mentioned the handling robot with the high throughput. Right next to it are two HPLC machines, Y2, because we can use more buffers to be equilibrated or collecting. So up to 8 buffers are available. This means every users have own buffer, and this allowed us to collect 8 users in continuous way that we do not interrupt the data collection. We also have a multi-angle light scattering and fluctuating this, that adding additional detectors to our collection that we can calculate molecular weight, hydrodynamic radius, etc. This was just mentioned how Greg collecting or recollecting high throughput SAXS using this needle/sample cell. We developed some kind of stage that SEC-SAXS is part of it, and the flow cells is built not from the mica, from the capillary, but from the mica window to lower the background. These are technical details that I want to not go into details now. Obviously, thanks to Daniel, will work on this. So what is the CAPL SEC-SAXS? You already have seen the talk from Steve. I will just go more specific for SIBYLS. We require about 50 microliter sample. The concentration, depending on the size, obviously, of your protein of interest, is about, I would say, 3 to 10 mg per ml. If you have a smaller particle, you need to have a higher concentration because of dilution at the SEC. And so that is not only the end of the data collection, we're collecting also multi-angle light scattering, and why it is important. So this is an example when obviously we have a nice single peak from size exclusion, and because we're collecting the both SAXS and moles, we can overlay those detectors and see that across the size exclusion elution profile or size exclusion elution peak, so for each frame that is collected every 2 seconds, you see the radius saturation drop from about 35 to 28 for this particular protein. However, we look at the molecular weight, that is more or less stable across the elution peak. So that's a really surprising. And so what is really happening in this example, why it is important to collect both. This is a protein, ASPP and PP1. It's a protein complex that have a multiple interaction in between. And so crystal structure was solved. And when we look at the crystal structure unit cell, there is multiple complexes in the unit cell. So that's the movement, what we see in the crystal structure. However, this crystal structure is not fitting our data, neither from the peak or from the side of the peak. So in reality, the size exclusion, the complex in the solution have much more bigger motions, and because of the transient interaction of this extended linkers between those two parts. And so this ratio between compact and open structure definitely changed if we are fitting the data at the peak or fitting the SAXS profile at the shoulder of the elution peak. And that's kind of confirmed that we can separate these different opening structures at the size exclusion. Specific case where the transition between this open and close is controlled by multiple interactions in between those two domains. this was just example why this is important. So what I mentioned, we have this 8 buffers, and as we are collecting, as we're collecting one sample, one column that you can select at our mail-in page, then the other columns are equilibrated, that we're saving the time between the buffers, between the users. Again, collecting the both small description and SAXS. And our mailing page, you have information about what columns you can select. You also need to add information about the sample name, and the minimal information. What we need is the volume that you are shipping to us. If you add a little bit comment on it, that's also great for us, or concentration at least, what you expect to have in your sample. It will allow us to get you a little bit more information. If, for example, you expecting it's a dimer, we will most likely delivering you much more bigger package of the results, not only the data or merge data, but potentially we'll be receiving also information about the molecular weight estimated from SAXS moles, PRL functions, and the models calculated the dense that you're seeing and a Thomas stalk. And when you add some information about this phase durable symmetry that you're expecting, the organization said you can add that in calculation and deliver to your higher symmetry models. Well, this is how is the flow done. You have the mailing page, when you put your slot, you define which column you have, how many samples, the name, etc., etc. This is download directly to a GUI that Daniel already mentioned, and that's why it is important, this information for us, that you give us some information about the name and the volume. It goes straight to our GUI that started all the process of data collection for moles and SAXS. And finally, Greg was already talking about the simple SAXS database. We believe up to next week when we're starting, we will use this for the primary derivative of data. If you are not signed in on the simple SAXS, you will get the note from Catherine. Please sign in. The data will be available after you sign in for the simple SAXS. Just technical details how we're doing it, but this is more or less what you don't see. That's how we are operating and see the data collection in the real time. Yeah, we'll skip this one. We have mentioned a couple of times as SEC-SAXS, very powerful tool, obviously, for the users. They may come on a site and spend some time with us. We can collaborate with the SPR for affinity, like for this one. It will be probably important to see what affinity between ASP and BP1 is indeed with the DNA protein complexes as well. Some kind of general information when you receiving our merge data and your package about data quality, etc. You also are welcome to reanalyze the data, as was already kind of shown by Steve yesterday. We have the package also include this SEC-SAXS profile and have all the frames together in so HDF5 file format, and you can directly upload it to the raw. I will give you an example in a minute. There are some issues that we mentioned a couple of times, like baseline shift. You can accommodate it and mathematically correct for baseline shift, or also Steve mentioned the baseline shift through the capillary flow. And this is very important to eliminate these artifacts from capillary flowing or not perfectly equilibrated columns like the baseline shift here. You don't see it in the moles data, in the moles for exclusion, but you obviously see it in the SAXS data. That's this bottom, the top panels versus the bottom. And really the buffer selection is also very important. Like example here, when you select the buffer that is not affected with the capillary flowing, give you a different profile versus the buffer that is selected after the evolution profile. Also, I forget to mention, you guys have a question, just interrupt me. Raise the voice. Idea was mentioned also to eliminate completely this capillary folding. We play with the idea of using the co-flow. We worry about that the high throughput SEC-SAXS will kind of lacking the speed, so we still negotiating with this. So we find a way when we're running a standard protein before your samples, we kind of slightly eliminating the capillary folding and the artifact in this baseline shift, as you can see here on the panel. We also allow to users to send own columns. So that we have cases when our column, the biggest what we use is 804 SHOTX. That is about for 1.2, 1.3 megadalton particles. That's maybe too small for you. You have a really large particle. We're working with Carolfeld on some idea about large assemblies like synthetic bacterial microcompartments. Those are 3, 4, 5, even 10 megadaltons large size. We coming with buying some new columns. They have the potential to separate very large particle, and some initial results have been done about 3.5 megadalton kind of virus stuff type of capsid that we are able to separate from the aggregates and receive very nice SAXS data, and also calculate potential parameters like PR function, etc. So that's the future development. What was done, and I want to mention this one again, how not only few samples can be collected, but in collaboration with Patrick Shi, particularly Albert Liu, who's a PhD student in Patrick Shi lab, we collect about 30, 40 SEC-SAXS samples of different forms of rubiscos. And it was done mostly overnight. We can run 15, 20 samples of 15 samples overnight. So it is getting possibility to run also your different mutations or different constructs. It's much more difficult for us to run the different buffer conditions because we need to all the time equilibrate, and equilibration is the time-consuming step in a SEC-SAXS. But if you have a different mutants constructs for different proteins or complexations, if you're interested, please, you're welcome to send more than 2, 3 samples. So far, the limit is about 7, I believe, at our mailing page to give in one slot. If you want to have more, yeah, just ask Catherine and we'll discuss the possibility to give you 2, 3 slots. I mean, there's no limit if there is open slots for the users. Just how now, before I go to the example, how to analyze, or just repeat what already Steve present nice way, and I wasn't prepared that it will be such a good tutorial from his side, so I just prepare another kind of example that we can work together if you would like, and also have uploaded raw. That's what you need. I will put in a chat a link to a data set that we can go through. But before I go there, this is example what was mentioned by Dina, that we can use this kind of conformational sampling on multi-domain proteins, or kind of looking for allostery and matching the SAXS data with the multi-states model. And again, we already have this view that every piece of structure, including glycans, ligands, are corresponding, are in our SAXS data. So it's important to model all these species together. It's an example from Bettina that she's staying with us for 6 months, and she was analyzing this protein that seems to be have a lot of glycan, and on top of it, emotions between this heme and FAD domain responsible for electron transfer. And so we are using BioSAXS in this case because it's much more complex that we can use in the multifox, but the multifox is obviously implemented to calculate this multi-state model, where you see the goodness of fit of chi-square for one single model, even that is the best fit model is about 2 point something. And when you're adding the conformers, you potentially improve the match to the data. You see it in the residuals when you use a multi-stage model. I think in this case, there's 3 models that are shown here on the right side. And you can use this kind of modeling tools to look at the different, in this case, Bettina was looking at different pH, like how this flexibility got affected with the pH, where the lower pH, you see much more compact conformers. The frequency of compact conformers is higher at the lower RG versus at the higher pH. You see the models that have been selected using Fox, multi-Fox are have a higher RG. That's allowed you to kind of say, you can see this already in the PRR function, you can see it in relative durations, but this is kind of additional information, how really those domains are separated. And so now example for SEC-SAXS before I go to a little bit more tutorial. I think I have a few minutes left. Over the pandemic, Beamline was kind of not closed. We have a minimal operation on our side. Thanks, Daniel. He was collecting data, all of us sitting at home and analyzing it. And so there was a great project for replication transcription complex that is a huge complex of multiple pieces. They call it NSP12 is the polymerase, where 7 and 8 are the cofactor. And indeed, the SEC-coupled SAXS with the MOS was crucial to really separate these transient complexes and find what's really happening. For example, here you see NSP12 by itself. When you're adding 8, is adding the molecular weight. Would we find out that this is not binding RNA, but it's only one piece of 8. The cofactor is on a polymerase. It's all changed when you're adding number 7 there. This is this orange one, the very tiny one. You bring the second NSP8 and also bind RNA, and that is a very stable complex that you can see here. But obviously, to model this data from the SAXS and kind of matching this cryo-EM structures and find out the transient complexation and what is important to the assembly of this large transcription complex, that cytoskeletons of the SAXS allowed us to do it, because we separate all those non-complex state, like you can see here from the complex. We still have pieces of non-complex states in the sample. This is an example where we go. And before, I think this is the last slide about a little bit modeling. I know I'm going back and forth, but again, to model the structures, solution state, I think the SEC-SAXS really allowed you to believe in your data and pushing the limits, like with the AlphaFold, for example. Now, AlphaFold have a good sense for fold, but unfortunately, the overall topology for much more multimodular structures there is lacking. And so here, give you an example of like protein for XSEC1, and AlphaFold recognize there are 3 folded regions, so N-termini, VLCT domain 1 and 2. And it seems like based on this PRDT matrix, there is kind of a probability of fold, or expected position error between aligned residues. There are some non-folded regions, but most likely flexible. But still, the AlphaFold come up with some very compact topology between those 3 domains. And so what we are doing, you can use the multi-Fox web up and just adding those residues as linkers, or you can also using the Bilbo ND that is part of our web app. You can just look at it right now. And we are able to assign the flexibility in a much more interactive way at our web app application based on this PLDT matrices that you can say, this is your alpha mode prediction but obviously you can just see that this region here, 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 and also here, this seems to be completely unfolded. And so when you're running BioSAXS, you come up with some models. This is atomistic model shown in kind of envelope resolution, but it's atomistic models. And so on top of it, the SEC-coupled SAXS show that this protein is not in monomeric state. But also from the molecular weight, we see it's not in dimeric state. And from the, I don't have picture here, but the broadening of the peak, and we will show in our tutorial that's exactly happened for lots of protein states, that this is monomer dimer mixture. And obviously you're gonna run BioSAXS of monomer, you're gonna run BioSAXS on a dimer because we know where the interface is. And using the multifox, can use also the web-app multifox, Fox multifox. You can see that it's a mixture of monomer dimers where this mixture nicely match our SAXS data, and the SAXS data match is here shown in the real space. Okay, so if there is not kind of specific question for this work, I would like to go to show you what is possible with the raw. I know it could be repetition from Steve talk, but I will put a link for a data set that you can work with me. It's not necessary. Here's the chat. Here's the chat. Okay. Like this. So much Windows, I apologize for it. So as you go to SIBYLS, now let's go to SIBYLS here. This is SIBYLS, and then you can click on a dashboard and you will see multiple codes. When you're looking for NSPA, that's the data that we're going to analyze. There's a couple of NSPA data sets, but for the first one, they are including also some models. So I give you this link here at the chat. You just can click directly and go straight there. I say all. Okay, so that's NSP8 itself without RNA, without DNA. And in the SIBYLS, I hope you guys see it. Here you have a link to the raw data sets. Now let's just start with this one, and you can upload it by just clicking data sets. And when you open the zip file, you will see that there is, unfortunately, there is also inside this .file what is the final merge data. But what you are interested in, you wanna really analyze it in raw are those 1 to 600 frames. So those are the single frames for our elution. Every frame is a 2-second exposure across your elution. And so let's just start the raw. And if you start the raw, you can click here on a series, and it's right here too. I'm gonna start the raw from scratch, so we're gonna close it. We can just follow up. So we can start the raw. Okay. Okay. So here we have our raw. So you can upload the data here. I'm a lazy guy, so I just click here on the series, and raw allows you to just grab the data set and put it in the series stuff. And he will give you automatically this kind of SEC-SAXS profile. What I also recommend before you look at the SAXS data, you look at the MOLS data report that we're also sending out. So here is the report for the MOLS data. So you will kind of get this kind of bunch of data sets that are the raw images. Okay, but you also get PDF file and README file that you know what this means what. And so there is so-called NSP MOLS PDF file, which is a report in the PDF format. Most more important is to look at that, just give you initial idea what is the quality of your data sets. And so when we go here, a little bit smaller, but these are the all light scattering, UV, and reflectivity index align on each other. So you see you may have a little bit aggregations. That's the light scattering C first, obviously, and then the UV, reflectivity index, and light scattering have one nice peak. And so this is dynamic light scattering signal, so-called quals. And then you get information about the molecular weights. It's not so good in here, but you can see better here. The molecular weight, so this is a little bit truncated here, but kind of you get idea across the maximum of the peak. The molecular weight is 36 kilodalton, and it's dropping to 34 at the peak. That's unusual. I mean, that's kind of surprising me because this NSP8 is supposed to be 22 kilodalton. So I'm already expecting some strange stuff going on. That's the first information. But let's look at the SAXS data and just make a SAXS assignment. What is the molecular weight? I prefer just a little bit. This is integrated intensity across the whole scattering profiles that we are giving to you. Unfortunately, we try to go as close to the beam stop. So there's a lots of noise at the first 3-4 points at the small Q. And so sometimes to see it nicely, you can go intensity in a Q range for the Y-X, and you can type, I want to see the integrated intensity between 0.01 and 1. So then it's kind of smooth up a little bit the curve. And you can see the background have also a little bit issue, but it's a very nice peak. And how we're going to analyze it, the next step. If you click on this series here, you already, Steve mentioned, you can go to LC series analysis. And so here, what you see are the same what you've seen before, but now we're going to need to adjust first the buffer. So let's say I want to add the region of buffer. And yes, obviously, the buffer here in front of the peak is the best selection. This peak, the buffer could be affected by some small molecules here at the side. I don't have the problem here. You can see the moles. There is no deep or anything that's a little bit far away, but still, to be sure, just pick up the buffer from here to here, set the buffer, and continue. The next, what I see here is a little bit capillary folding, very minimal, but to be sure that we are not including it. So we do baseline correction, and we click the linear and do baseline correction. I personally think that you can keep it this way, but if you want to be sure, this is kind of a void here. That's a little bit aggregations. We're just gonna have a linear adjustment here and set the baseline and calculate. Some complaining, ignore that. And you see the baseline is much more smoother now across the peak. Well, the next step will be basically at the region for the sample, and that's very important. Okay, to zoom in, and before I go there, and select the peak. But what this is very informative as well. So already you see that not only the molecular weight is dropping across the peak, but also the radial duration. So this is radial duration about, sorry for the last outlier here, but about 32 to 24. And these are definitely significant changes in radial duration across the peak. You can also look at the molecular weight. So by changing here calculated values, you will see what are calculated molecular weights for each frame. And it's very similar. So this changing the radial duration reflecting also a different oligomerization state, because you see the molecular weight change from 34, that's what we see in the molecular weight, to almost 22. You remember I told you this NSP8 at the monomeric state is 22 kilodaltons. So it seems like only those last tail of the evolution have mostly the monomeric state, versus here it's something in between monomer and dimer. So already from this information we get, you kind of get an idea how the sample behave. So when we add the region and select the sample, we'll try to do it. Okay, I'll have it run, have it. Okay, let's do it again a little bit better in the zoom in window. Let's just take just the peak with the highest molecular weight, and we can set it to okay. Continue. Well, let's also select the size of the peak, and we add the region, and select just the size where the mostly monomeric state is. So just looking closely on it. Okay, just make it a little better selection here. We need at least some signal, because here is already no signal at all, so there will be no SAXS, obviously. And to profile. And now we can kind of ignore this part. And when you want to see what's your SAXS profile, you go here to this profile. Just look at those curves. What is the next step? What I will recommend to do is just, when you go here, you see that's the peak and that's the size of the peak. You definitely want to calculate the different, and you're getting this information from us anyway, but it's good to redo what the busy Beamline scientists do for you. It's a calculate the Guinea, okay, and using the IPI, what I mentioned, it will be done automatically, so it will be not so much work. You can do the Guinea for the size of the feet, the shoulder, and I will also definitely calculate the molecular weight. And you get the multiple calculator here, and you see the molecular weight is something in between 33 and 37. 34.6 is based on Bayesian, and then the different calculators here as well, like for the correlation and for volume, or based on shape and size. When you do here the same molecular weight calculations for other sides of the peak, okay, I forget to calculate the Guinea. You can't forget to calculate the Guinea for it, and you need to click on OK. We don't do it. And you see the molecular weight drop a little bit instead of 34 and 33, but it's still pretty high. And if you want to compare the values that you just did, you can just clicking on those files here, and you see the RGs here, molecular weights down here, and same for RGs here, drop a little bit, molecular weights are dropping as well for this shoulder. You've got some idea about how this behave, and now what we can do, and I will do it very fast way, but let's just take the hoping the most monomeric state, that's the orange one. But let's just calculate the P raw function and compare those. What's really changed? How is this monomer dimer mixture versus more monomer? How the P raw function changed? And that was already shown by Steve. I'm gonna repeat myself or repeat his presentation. And if you have an installed AXA package on your computer, you can kind of easily click here on GNOME, and we will bring up the GNOME, or like everybody say GNOME, but I prefer GNOME because it's made in Germany, and they don't say GNOME, they say GNOME. So this is a nice P raw function, go to the 0 about 115, and it's kind of artifacts here, but I think was excluded. You can also help this do a little bit more by taking viewpoints from the beginning, but all well together. It's okay, and the raw function is saved. You can also calculate the raw function for the shoulder of the feet. Okay, this is again a little bit beginning. We'll just truncated these few points here. Why not? If the particle is relatively small, you don't need those points. And now we say, okay, again, when you go to top in EFT, you will see the comparison that indeed the shoulder is a little bit smaller in the Dmax, and is also less broader. And these are the fit from the GNOME. But in generally speaking, what was recommended using this deconvolution, and let's try it. I know it's not going to work for this case because this transient behavior where monomer dimer is appearing across the whole elution. So there is not a perfect separation between this monomer and dimer. It's really just a little bit. You can see in almost similar PR function and almost similar radius duration, and only a slightly difference of molecular weight. So if you go back to your OC analysis and you decided to do something more fancy, okay, we just click OK here, we don't need this now. But when you go again to the series, it's right here, okay. Again, you have options to SVD or legals or EFA. The EFA was mentioned today as a tool when you want to deconvolute, and we do SVD at the beginning and immediately recognize there are two components. One is very major, and there's a third component that is really minimal, but let's just assume there are two components to make the system a little bit simple, 1 and 2, and so just ignoring those 0 components. Well, if you do very automatic next button, kind of show you where the second component is barely rising from the background. And when you do next, you will see that you will not able kind of separate those. You can slightly adjust that region of second peak, make it a little bit bigger. Well, it's very questionable if this is a good solution. I mean, based on chi-square, we will kind of believe yes, but if you really want to minimize the chi-square fit of these two curves to the entire set of data sets, you will realize you will not able to separate those almost. Yeah. So you see, then almost impossible. So I know that this is frustrating, but it's not working for all cases. So you need to be careful when you're applying EFA. But there is a good news here. So we say cancel this. And let's go to our profiles, and we need to save those profile and do some exercise with those scheduling profiles. And we're gonna save those first. Here's the button save. Well, I can put it here, why not? Call it MSP2 data. And this one we're gonna say shoulder. Okay. And in the meantime, when we're gonna work a little bit with these two data sets or two curves, one from the feet, one from the shoulder, I will show you how easy it is to really go to calculating all these shapes, because even that you don't need it because we know the structure, and I also show you how easy it is to run AlphaFold on this sequence. So basically, by going to IFT, what is the GNOME outputs? Let's decide to take this shoulder, and we're gonna save that too, but we don't need to. You can go here or even here. It's called electron density, and again, what I really appreciate, our speakers show this tools pretty much in details. Just give you an example for this one. For cost of the time, I will not start it, but it's take about, oh, why not? We're gonna put it in a fast mode, and we're gonna calculate only 5 reconstructions. Yeah, and I know it is almost a monomer with about 20 something, but I don't apply the symmetry in this case. I will keep just the 5 reconstruction running in a fast mode, and I will not do refinement because it takes a little bit longer time. And I will just have only one CPU just in case you don't have multiple CPUs. Okay, so no, I will change this name. Sorry, I need to change the name here. Okay. And now we can, okay, I'm sorry. We just do 5 models. And in the fast mode, no refinement, just a version. And it's start. So it's gonna take about, in this fast mode, about 5 minutes. I did it before, and we'll just fire up Chimera. Okay, kind of ignore what is here. It's already done. Okay, here's my Chimera. Okay, I'm a big fan of Chimera. This is Chimera X. I just started to use it like a couple of days ago. So, but basically it's a new Chimera. It's much more user-friendly. And let's just so we're gonna upload the MRC file. You will receive an MRC file for each run for 5, but obviously you want to look at and drop, and you will see that your MRC file, the final calculation. Obviously we want to put it a little bit in. So here I have the Chimera also have a structured prediction. So if you have a sequence that you want to predict, show or not show something. work with that. There we go. That's what I was looking for. This is your shape. This is your initial shape calculated from bands, and obviously you already seen how you can adjust it to the sigma that you want to look at. This is what you see here in the ribbon. That's just over when Greg was talking, I was calculating this alpha fold model, sorry. And the alpha fold model is basically also shown in the colors where the low probability fold is in yellow and the blue, and it's so they make it so easy that this Google interface or the Google lock is directly plugged in a camera. So if you go here, okay, I'm going here. Yes, when you have a sequence uploaded, I already have it open. So, here we go, tools. And you have the possibility to have a tools selecting as alpha fold that you directly are linked. And when you have a sequence, you can go to tools here and say alpha fold calculation. And now my alpha fold plugin is already open. So it's right here. So you basically put the sequence here. I will make this window a little bit bigger for you that you can see what else plug in different places. And it took me 22 minutes. So you plug the sequence here, and play with the sequence alignment or just completely automatic modeling. And after 22 minutes, you come up with this model of NSP8 with also the PLDT matrix, and you kind of see where are correlating with this low fold probability colors, where you kind of also have the same visual here. And so we have this alpha fold, and what's the problem is in alpha fold is that, again, based on a sequence alignment, and what we already immediately can see that are ab initio shape and also radial duration and any kind of indicator from the SAXS, we kind of disagree with the alpha fold. And what's really the only issue in this case is the flexibility and folded back structure in this coil region. And so again, you can use multifold or Gilbert MD. You can take this model, what you kind of calculate in 22 minutes, and do some modeling. I don't have a time for it right now, but instead of this alpha fold model, you will receive based on your fit of the data, you feeding your SAXS data directly. You will get this model here. I'm gonna disable the shape, and you will see that what's really happening here is just basically this local ability and flexible region allowed the folded back structure, and actually this this model fit data, SAXS data pretty nicely. And not only the SAXS data, but I know obviously you are keen to see something in 3D. This is a very fast 5 runs of dance, or it's definitely improved if you're doing in slow mode. and doing at least 210 models, then you can get a better agreement between this folded back NSPA structure. And on top of it, as I already mentioned, there is a problem because the Hammel molecular weight is a little bit higher. So it's not only monomeric states. So why we're not matching this blobs here? Because there is some contribution of this dimeric state. And to be sure about this monomer dimer transition, again, Dina mentioned that, so I will show you an example what to do with the Fox. So when you go to the Fox, and please don't go to the Fox News, you just go BioSAXS, give you a Fox server. In the package that is in the simple SAXS, there is the raw data, but also there are link and models, obviously the moles report as well. But then you have there, you want to try it with me, you can now upload this PDB called dimer compact tool and this other PDB, what is the monomeric PDB that I'm showing you now in Chimera. And if you upload them together to the Fox, and Dina didn't really mention that, but I want to do it now. So let's just do that together. We have here, this is a different workshop, unfortunately. Here we go. And these are the whole package. Here I have those PDBs number 1 and number 2. Those are the PDBs are available online. I will zip them together. Okay, and produce a zip file that can be directly plugged in to our box. That up, and we're gonna take the dot file. Let's just do the dot file that we just produced. And I need to look for that. Let me create it. And maybe I didn't save it. Save it somewhere. Oh, let's go back and see if I save it somewhere. Here's my profiles, then I save it. But we didn't save it. Okay. Yeah, it should be there. Okay, let's go back. I need to be somewhere in my directory. Okay, so here are here it is. Shoulder. Shoulder dots. 3 and shoulder out. Here we go. That's my dot file of the shoulder. And I can take that to the experiment. And when you click submit, you will have directly a fit for those 2 files. One is the monomer and second is the dimer. The dimer is from the crystal structure of a homolog. It's not exactly the same COVID-19 NSPA. It's a very similar coronavirus. And you kind of already see the monomeric state, the compact structure fit the data almost nicely, but there is overfitting with this parameters called hydration layer, C2. And so when you hit the multi-state, that you'll fit improve from 2 to 1.1, what is almost perfect. And also hydration layer became a little bit more normal. And the most important information is here. We will have all the residuals improve, the fit is better, but mostly also you can see the weights that you still, even at the shoulder, you have a few percentage of this dimeric state. It will be interesting just compare that ratio between monomer dimer for the peak data. So instead of NSP8 shoulder, we're gonna take NSP8 and do exactly the same exercise. This somehow give you an idea how we can, in a fast way, obviously you wanna get rid of those frequency or the beginning for the time. Do it. Yes, in both cases, we improve the chi-square by adding a mixture of, in this case, it was about 80, very similar, 80% and 90, 20%, 81% and 19% of mixture of monomer and dimer. And most important was really improve the small Q region. So you can go here, there is a significant improvement by using a few percentage of the dimer. Yeah, in here, so in a high Q region, so that's a bit better. So this is just a fast way to validate the mixture that you see in SEC-SAXS / moles data sets. And again, with this impressively fast way calculate atomistic model in a relatively in a resolution that is good enough for solution scattering, as I showed here. You kind of start to validate your SAXS data not with the blobology, but more likely with more constrained using the atomistic model, combining cryptostructures or predictions. I think that's as much as I can say right now.